Turner's presentation of her dissertation proposal. She will be presenting her topic, Evaluating Intestinal Microbiome Dynamics Across Perturbations to Predict Treatment Response in Chronic Inflammation. Christina has a background in molecular biology and microbial ecology. She views the human as an ecosystem, and her main interest is to understand the relationship between microbes and host. Christina hopes to get back to doing some wet bench work once she completes her PhD. She has 2 dogs that keep her company while she works from home, and they've been exploring various parks and trails in the Vancouver area. I just want to give a little outline of what's going to happen today. Christina is going to present. Then there'll be opportunity for all of you to ask questions. And when questions complete, those of us who are not on the committee will step off of the WebEx, and Christina will stay with her committee for any final discussion. And with that, I turn over the presentation to Christina.
Okay, thanks, Karen. And I made sure to feed the dogs an early dinner so they don't harass me while I'm trying to give this. And I see that it's recording. Okay, so as Karen said, my topic is on looking at intestinal microbiome changes or dynamics across perturbations to predict treatment response in chronic inflammatory conditions. And to best answer this, I plan to take a salutogenic view or approach. And this model of health views health as this process along a continuum, rather than the more traditional pathogenesis view, which really looks at health by trying to understand one disease, one treatment approach. And cytogenesis also views the human as a system with interconnected subsystems. And one of these subsystems is the microbiome. Should change. There we go. Okay. So down here you can see one of the subsystems, the microbiome, and the other subsystems are depicted along this outer circle. And these can be affected over time by factors such as stress, your genetics, and your lifestyle. And these factors can lead to a health outcome that affects like your blood pressure or your temperature or glucose levels. One second. Sorry, one second. Okay, that works. And in regard to the health of our gut microbiome, we know a lot about the mechanism behind chronic inflammation and its association with an imbalanced or dysbiotic gut microbiome. So certain chronic inflammatory conditions such as cancer, Alzheimer's disease, inflammatory bowel disease, and obesity are known to be associated with this imbalanced microbiome. And that can be depicted in this right image shows kind of a lack of diversity. So there's not as many different colors as this healthy gut. And what happens in this dysbiotic gut microbiome condition is that the intestinal lining barrier loses integrity, and the toxins and byproducts that are supposed to stay in the luminal space can cross that barrier and enter your bloodstream. And this can cause endotoxemia, and constant endotoxemia leads to chronic inflammation. And I do want to define what I mean by gut microbiome. I'm just saying gut microbiome in GM, but I'm referring to the intestinal microbiome, which are the microbes, including like bacteria, viruses, fungi, and archaea that live in our intestine and on our intestinal wall. So given this link between gut microbiome dysbiosis, chronic inflammation, and the pathogenesis of chronic inflammation, I'll look at chronic inflammation as kind of the disease model to understand this overarching question of how does the dynamics or changes of the gut microbiome impact host health. And this really looks at gut microbiome health along a trajectory of chronic inflammatory conditions. You can see here, these outer boxes represent different conditions that are associated with a dysbiotic intestinal microbiome. The ultimate or long-term goal of this is to prevent or reverse the pathogenesis using the information from the gut microbiome. And a more closer goal would be to identify indicators of risk for dysbiosis, so more about early detection and prevention of dysbiosis. And this will allow us to give us potential for improving or developing more microbiota medicine, so like probiotic interventions or diet, or even like fecal microbiome transplant. And one chronic inflammatory condition that is well studied and known to be closely associated with gut microbiome dysbiosis is ulcerative colitis, which is a subtype of inflammatory bowel disease, and it's a complex chronic inflammatory condition described by an inflamed colon. And this diagram was from Clemente et al. That did a systematic review looking at different studies that tested perturbations or treatment interventions on mice and human models of colitis. The perturbations they looked at include diet, probiotics, antibiotics, and fecal microbiome transplant. And they found that there's a shared trend with increased bacteria or decreased bacteria associated with ulcerative colitis versus a healthy control. And so blue is more abundant, red is decreased abundance. They also found that microbial byproducts such as short chain fatty acids are decreased in colitis conditions, and then other immune markers are also affected. A approach that I propose to take that's a little novel is to apply this concept of resilience to understand this relationship between gut microbiome dynamics and chronic inflammation. And this concept of resilience can be explained with this ball and basin diagram. You can see one state here, state A, which is represented as like a non-inflamed gut, a balanced gut, a happy human. And then this state is an inflamed gut with a dysbiotic or imbalanced microbiome. And the system, which is the ball, what can happen if the resilience is low and the perturbation is strong and frequent enough, the perturbation can push the system across its threshold or tipping point and cause a critical transition from state A into state B. So that's kind of the concept of resilience. And the width and basin of this diagram represents how resilient the system is. So the state A is less resilient, state B is more resilient because of that depth and how narrow it is. So one of the challenges of this concept is that this term resilience is complex and it's multifaceted. It's also a very context-dependent term. So for instance, this concept of resilience has been applied to many other disciplines, so such as like brain health or even like soil systems or lake systems. And over the next few slides, I'm going to discuss more about how I'm going to break this term down so that I can actually apply it and understand how it relates to the gut microbiome, the human gut microbiome. So to apply the microbiome features to resilience, I'm going to first talk about what I mean by microbiome features. Species diversity is the number of different species in an ecosystem and the relative abundance of each species. And you can kind of see that from these pie charts over here. The different colors represent the number of different species and the abundance. And then taxonomy or composition refers to the bacterial relative abundance. Functional potential or relative abundance refers to the bacterial gene family relative abundance. So in other words, taxonomy is the type of bacteria, function or functional potential is the what the microbes are doing potentially. And I've selected these features because diversity has been found to be associated with healthy hosts. So a greater diversity, more healthy hosts. And then with taxa and function, kind of like the previous ulcerative colitis diagram I showed, there are particular taxa and functions that are associated with increased inflammation or decreased inflammation. And then applying these features to resilience, similar to the ball and basin diagram I just showed, we have a community of bacteria here, like a sample. in state 1. And then the idea is this, does this shift in response to perturbation to a second state? And the idea is that when the community shifts from state 1 to state 2, does the diversity, taxonomy, or functional potential change? And if it does, how much does it change? What are the features that explain that change? So one of the challenges, as I mentioned, with resilience is that it's a complex term, and there's also no unified or validated definition to kind of measure resilience for the human gut microbiome. So for my project, I propose to focus on just one component of resilience to get one step closer to being able to characterize gut microbiome resilience and host health. And so the component I plan to look at is recovery. And the definition of resilience that I've adopted from Sheffer et al. Is the system's capacity to bounce back to normal, healthy baseline functioning after a perturbation. And the two key components of this resilience definition is the resistance, which is the degree of change the system makes after perturbation, and then recovery, which is the system's return to baseline after the perturbation. So I plan to look at recovery. And then these two diagrams kind of depict the recovery in a high resilience system and a low resilience system. The idea here is that the faster the recovery, a faster recovery is more indicative of a system with greater resilience, and that's shown with a deeper basin has a steeper slope. So the system kind of rolls or returns to that equilibrium faster. So it has a higher recovery rate versus a low resilient system has that more shallow basin. So the slope is less steep. So it has a slower recovery rate or low recovery rate. And I do want to point out a key consideration here is the rate. I will be able to look at microbiome features that are indicative of recovery, but not necessarily informative of the rate. I will be able to interpret how time point distributions affect microbiome features though. So the ultimate goal is to understand the utility or use of the gut microbiome dynamics across these perturbations to assess host health and treatment response in chronic inflammation. And I propose to do this with the following aims. So the first one is to evaluate perturbation dynamics of the gut microbiome in healthy longitudinal non-perturbed and perturbed data sets. And this will allow us to determine if these dynamics are informative of health. Second, I will build a predictive model to predict treatment outcomes in different chronic inflammatory conditions based on pre-perturbation or baseline microbiome features. This will help us understand the features that are more informative of gut microbiome response to perturbation. And I hypothesize that there will be a subset of features that are indicative of post-perturbation gut microbiome changes, and also that are predictive of host treatment response in the multiple chronic inflammatory conditions. So to start, I'll talk about aim 1, evaluating the use of perturbation dynamics. And the main question here is, are the longitudinal dynamics informative for assessing the ability to return to a baseline? And this question was inspired by the study by Olson et al. That looked at temporal changes in bacterial abundance in healthy adults. They found that the intra-individual variation does stratify in healthy adults. And this is just a graphical representation summarizing their main finding. They were able to categorize the temporal trends they saw in these non-perturbation healthy individuals and found that there are species that categorize into stable, bimodal, and variable species. So then the question comes up of like, if we see these in non-perturbation temporal trains, how do these trends look in a perturbation gut microbiome response in healthy? So then for the approach, I'll be using independent publicly available data sets of longitudinal and perturbed time series of healthy human fecal metagenomic data. And this will be to identify significantly different taxa and functions and measure the variance between time points across each time series. And so then I can compare them across these cohorts of normal and perturbed healthy adults. Trying to show a conceptual representation of this. Here's a normal gut microbiome time series, and at each time point, I'll have either the taxonomic relative abundance distribution or the functional relative abundance distribution. And then here's a nice lightning bolt with a perturbation. So this shows a perturbation time series. And the microbiome features I'd be measuring is the alpha diversity, which is the variation within samples, the taxonomic and functional relative abundance. Sorry. And the between the time points to identify which samples are significantly different in community composition and function, I'll be doing a longitudinal differential analysis, differential abundance analysis between the time points. And that will also allow me to identify any key taxa or functions that explain the difference between each time point. And then I can compare those between normal and perturbed. To measure the variance between time points and get the delta, I'll use the Bray-Curtis dissimilarity metric, which is a commonly used measure to assess how different microbial communities are. A little more details about the data sets I'll be using. I'll be using the healthy individuals from that Olson paper that found the trends in the temporal. And this non-perturbed time series has 4 time points per individual across a year. The second cohort is of a perturbation data set of antibiotics. And there's 10 individuals as control who were not given antibiotics. This was 23 time points across 4 months. The third cohort is also an antibiotic intervention in healthy individuals, only 5 individuals. This one had 11 time points across about a year. And then to show details of the time series, the non-perturbed time series, the time points were collected equally across the year, across 4 visits. The second cohort, the samples were collected during baseline, during the antibiotic treatment period, week follow-up, and then for 6 points for the remaining weeks of follow-up. So 6 sample points for follow-up. And then for the third cohort, there's 1 time point from before the antibiotic treatment, 4 during, which is the orange, and then 6 points follow-up. And you can see each individual kind of has some variation in the time between the time points. Between cohort 2 and 3, the antibiotics used was different. So that's something to consider. And then this aim will inform us of the potential utility of the post-perturbation gut microbiome as an indicator for host health. And it'll also help us understand if these temporal dynamics compared to the perturbation gut microdynamics are informative of healthy cohorts. And then for my second aim, I'll be creating a predictive model to predict treatment outcomes in multiple chronic inflammatory conditions. And the question here is, can we utilize a core set of baseline microbiome features to predict treatment response in different chronic inflammatory conditions? So different conditions or diseases that are promoted by chronic inflammation, such as like cancer, autoimmune disease, and metabolic syndrome will be looked at. And this question is really getting at whether gut microbiome features that are predictive of treatment response, if they're shared across different chronic conditions, chronic inflammatory conditions. And I'll use publicly available independent data sets of perturbation data. And I will be using the pre-perturbation time point microbiome features as input, and the outcome would be a binary treatment response. And I plan to use a random forest because of the variable importance aspect, and also random forest is good at handling high dimension data, which is microbiome data. And variable importance will be measured by how much removing a variable decreases the accuracy. There we go. So the disease perturbation data sets I'll be using are of, again, human fecal metagenomic data. And the conditions I'll be looking at include recurrent Clostridioides difficile infection with an antibiotic followed by a fecal microbiome transplant intervention. The other conditions include active ulcerative colitis, severe Crohn's disease, obesity, non-small cell lung cancer, and renal cell carcinoma, and then active ulcerative colitis again. And the other interventions include fecal microbiome transplant, surgical resection, a weight loss diet intervention, and then for the cancer cohorts, an anti-PD-1 followed by antibiotics or not, and then FMP. And as far as the responses, the outcome response defined in these data sets, they all define the outcome in a binary manner, but they use different disease metrics because they're all different conditions. So to simplify the treatment outcome for my purposes, I'm going to categorize these into success or failure treatment, so a positive or negative response. So for instance, the C. diff infection response, they defined it as a cure or recurrence. This one was clinical remission or no response. So for these situations, cure and clinical remission will be considered a positive or successful response versus recurrence or no response as a negative or failure of treatment. And then for building the model, I'll take these independent data sets of the conditions. I'll split them 70-30, and the 70% training sample will be combined, and then I will train the model and also do the leave one out cross-validation for tuning the hyperparameters. The random forest classifier will then be evaluated with the 30% holdout set from the combined data. And I'll evaluate the classifier using the measure of AUROC, the area under receiver operator curve. The model will then also be validated using the separate ulcerative colitis with fecal microbiome transplant perturbation data. And the predictors I'll be using for this will be coming from that pre-perturbation time point. So the first time point, and the microbiome measures will be alpha diversity, taxonomic, and functional relative abundance. And then also I'll be including clinical and demographic measures, including sex, age, BMI, and if possible, I'm going to see if I can categorize the disease severity into mild and severe. But I'm not quite sure if that's feasible yet. And then the outcome variables, kind of how like I mentioned, it will be binary, response or no response. So this AIM2 will allow us to identify the baseline microbiome features that are predictive of treatment response across these conditions, and to assess how general or specific these features are, how shared they are across the conditions. And then I'm going to go through my workflow. So I have a lot of different data sets that I'll be acquiring from different databases. and I'll be doing pre-processing them all the same. So I'll start with the raw metagenomic short reads. I'll do quality control and filtering. I'll normalize the reads, and then I'll also transform any data. I also forgot to include in this diagram that assessing the batch effect is really important for microbiome time series data, so I'll be doing that. The output from this will be the filtered sequencing reads, which I'll then assemble into longer sequences or contigs, which I can then annotate the taxonomy and function. So identify or measure the taxonomic and functional relative abundance. Also can calculate the Shannon's diversity or alpha diversity index. There's also 2 other indices that's been developed that, if I have time, I'm planning on assessing the gut microbiome health index and dysbiosis score. And these will be my microbiome features. And then I'll use that to input into my AIM-1 perturbation dynamics analysis step to do that differential analysis between time points to identify taxa and functions. And then I'll measure the delta between the time points and compare those between time series. So the output from AIM-1 will either be that the perturbation dynamics identify key taxa and functions that are more indicative of a perturbation rather than temporal changes. So if that's the case, then I will use that information to kind of supplement my model so that my model is based on taxa and functions that are more informative of a perturbation rather than just normal temporal dynamics. For the predictive modeling, as I said, I'll do a 70-30 split and train and test the model. I'll evaluate and then validate with the unseen ulcerative colitis FMT data. And then this will give me the feature variable importance and the model accuracy metrics. And even if AIM-1 output shows that the temporal dynamics and perturbation dynamics are not distinct, then I can still have information from AIM-1, and in the random forest model will be built from the microbiome features. So since my study uses secondary analyses, some current challenges with that is that I do have small sample sizes, which may lead to sampling and power issues. There's also a potential bias from using these different data sets that could come from different collection methods. The data sets that I'm using were also collected for a different purpose than what I'm doing, so that's also a consideration. However, the key goal is to provide a framework that can be utilized with other microbiome data sets and is agnostic of disease or tissue so that our impact is not limited to analysis of this data alone. So in conclusion, microbiome features that are predictive response to perturbation could be informative of treatment response in these different conditions, such as like probiotics or prebiotics, antibiotics, FMT, and also understanding the gut microbiome perturbation dynamics and assessing these common predictors can provide a foundation for future work to improve treatment efficacy and success in patients with chronic inflammation. And overall, the goal is to understand gut microbiome dynamics and health as a trajectory. And I say thank you to my committee members, Shannon McQueenie, Eilish Bajor, Michael Mooney, and Sean Gibbons. Also thanks to the faculty and staff of DMICE and my fellow students and friends, and to the T15 grant.
Thank you for that presentation. Let's now open it up for questions.
I think I definitely talked a lot faster than I thought I would, so we have a lot of time.
I have a question. I'm less familiar with the data sets that you're referring to. And I guess my question is, do you have any, do the data sets contain information about whether the patients were being treated or changed their diet or did something different that might have an impact on the gut microbiome?
Some of them do and some of them don't, which made it kind of tricky. Let me go back to the data sets.
So it's good that you have some that have those data because it seems like ultimately we would want to know how to coach patients and for clinicians to provide guidance on what to do when somebody has one of these conditions.
Yeah, it is interesting. Some data sets have like a very extensive like daily diary, and others don't. So to be able to kind of integrate these data sets and use them together, I'm just using microbiome features and pre and post perturbation. So yeah.
Thank you. Again, if you have questions, feel free to raise your hand or put a question in chat.
Hi, Christina. This is Nate. Can you hear me?
Yep.
You're quiet, Nate. Can you speak up?
Yeah, sure. So I'm curious on slide 26. Do you plan to use all the data sets to train the same model or different models?
Train the same models.
Okay.
Train the same model. Yeah. So I'll be training one model with 70% from each of the data sets.
Okay. Thank you.
I have another question. One of the tables you showed whether somebody was active or in remission. So I'm assuming you have some kind of a lab test or something that tells you when somebody is in clinical remission, like a calprotectin or something that would tell you whether someone's in remission or whether they're still actively inflamed.
Are you talking about this one with the response? Yeah. So each study defines that outcome different. But for instance, like with ulcerative colitis and Crohn's disease, they usually do like a colonoscopy,
right?
Or use like a Mayo score. So they all use a different health metric. But the study already defines like this patient responded or this patient did not respond.
So that means there'll be some variability across your data sets as to what a response is. So you'll have to account for that in some way, I guess.
Yeah. I think that's one of the perks of the random forest is the interpretability. So hopefully I can see, understand what's going on. Well, thank you for listening, everybody.
Christina, I actually have another comment and question. So if you're using all of these data sets in the same model, how are you going to control that the different, that essentially these different conditions may have very different unperturbed settings, and the model can use that to predict the outcomes, specifically for some of these data sets where there's a high class imbalance like that first one. Does that question make sense?
Yeah, you're asking like with the variability in conditions, like what if the microbiome features are really different? Is that what
Yeah. In each experiment, that the unperturbed microbiome conditions could be different, and so it seems like it could be hard to delineate response versus non-response versus, just predicting based on the experiment itself or the data set itself.
So the goal is to understand not necessarily what features are most predictive in the different conditions, but what features are shared across them. So if we find that there aren't that many features that are shared across the conditions, then that also tells us something. Does that make sense?
Okay, makes sense. Thank you.
Okay, I'm just going to give one more shout out. Anybody, if you have questions, feel free to speak up. Otherwise, we're going to close up the session from the public and let Christina and her committee meet. So let's give another minute or two to see if anybody else wants to ask a question. Very impressive presentation.
Thank you.
We have a question in the chat. Can you see it, Christina? It says,
Yeah.
how much concerned are you about sample size and power of the study? This is from Manish.
Yeah, that's a good question. I mean, it is a very preliminary assessment of these microbiome features associated with recovery and resilience. So I mean, it's a concern, but I'm still gonna do the study. But there are ways to evaluate the random forest model, and also with the AIM-1, the time series, and the perturbation. The idea is that I'm comparing the deltas and differences between time points so that I don't need to worry too much about the sample size.
Well, Christina, I have a question here. So it seems like you have a different type of diseases, right? See here you have 6 diseases. Do you expect the same set of features can be applied for different diseases in your random forest-based classifier?
Yes, because they're all known to be associated with the microbiome changes, and also maybe I'm not understanding your question correctly, but they're all the same data type, so I will be able to calculate those measures from them.
I think he's going at your actual hypothesis, Christina, which is, do you really believe that there will be commonality across all these diseases, right? Which is exactly what you're trying to assess.
Yeah, I think there will be, because there's a lot of taxa and functions that are associated with either promoting or inhibiting inflammation that we know about. So I think we will see those kind of trends across these different inflammatory conditions. So for instance, like that short chain fatty acid microbial byproduct that they know is decreased in ulcerative colitis.
Okay.
Yes, this guy, that's usually decreased in inflammatory conditions. And then other functional pathways such as LPS, lipopolysaccharide. I'm blanking on the name, but LPS is usually associated with greater inflammation. So I think there will be taxa and function that kind of distinguish that are identified.
This should you actually try to evaluate like C using a subset of disease, actually see the learned features they may be shared across subset of diseases or something like that.
So more kind of selecting diseases that are more similar to each other.
Yeah, or just to try to validate your hypothesis, or they actually shared all these features across all different diseases, right?
Yeah, and the idea with validating with this separate ulcerative colitis FMT data sets to kind of try to understand how generalizable those features are, but it is a small data set, so I could find other conditions to look at.
Okay, but in case your random forest classify performance is not good, so you may have some kind of alternative approach. See, I should really focus on subset of disease instead of try to average everything out, right? You may lose a lot of information.
Yeah, and I think for alternative approach, if the random forest really doesn't work well, I could try a simple linear model or like other models such as like support vector machines and kind of compare how those do.
Okay.
Christina, I'm not seeing any other questions posted or any other hands raised. We want to thank you for the presentation. And now I would ask that everyone, including myself, will be stepping off, and Christina and her committee will stay for any additional discussion. Well done.
Thank you.